Ladies and gentlemen, welcome to the Falling Walls Lab 2018, the second year of the country's most prolific scientific event.
Good evening, Your Excellency, honorable guests, dear colleagues and innovators, ladies and gentlemen. The story of Dorothy Metcalf Lindbergher, American astronaut, and our Falling Walls Lab presentation special guest 1 year ago is the story of the power of learning. Education can take you anywhere. She says she is a perfect example how far, even to the space.
Ladies and gentlemen, on the 9th of November 1989, the Berlin Wall fell. This world-changing event motivated quantum physicist Professor Jürgen Mlinek to organize a unique worldwide gathering of young scientists and innovators in Berlin. Starting from 2009, on the 20th anniversary of the event. Since then, young scientists and innovators are gathering every year in Berlin. Until 2017, without Slovak participation. In 2017, we have changed this. I want to thank all our young colleagues who will shortly present their results, ideas, and projects. I'm looking forward to all your presentations. And finally, I want to thank all of you in the Moises Hall for being here today, for supporting our young people, for supporting our future. Thank you for your attention.
So after all these introductory festive speeches, let's proceed to the business itself. We have the jury that was introduced to you a few moments ago. And before we essentially ask the first participant to present a presentation, I'd like to very briefly summarize the criteria according to which the jury will judge the presentations. So firstly is a breakthrough factor, and essentially is the originality and the innovation potential of the research reported. So after all this, I think that it's time to proceed, and I'd like to ask the master of the ceremony to start, to invite the first speaker. Thank you. And the first place goes to Lucia Lauková.
So good afternoon, dear ladies and gentlemen. I'm here because I would like to break the wall of sepsis. Sepsis is a life-threatening disease which is a major cause of morbidity and mortality worldwide, so it's a very big problem. Sepsis is mostly caused by bacterial infection, and this infection stimulates inflammation, and inflammation causes organ dysfunction and death. Standard treatment of sepsis is hospitalization and intravenous administration of antibiotics. These antibiotics kill bacteria. However, in a lot of cases, this is not enough because still a lot of people die from sepsis. Why it is like this, we don't know, but what we know is that bacteria are not the only present in the bloodstream. Normally, we have DNA in our cells, in the nucleus of our cells. But sometimes this DNA is released from the cell to the extracellular space, and this DNA is called extracellular DNA. And what we know is that in the blood of septic patient is a high concentration of extracellular DNA. And this could be a problem, because extracellular DNA is recognized by white blood cells and activate them and stimulate inflammation. And in inflammation, more and more DNA is released to the extracellular space, and this process is in the circle. And we wish to stop this circle, and our solution is DNase. DNase is an enzyme which cleaves DNA, degrade like this. So, no DNA, no activation, no inflammation, and this circle should stop. And this is what we exactly did. We induced sepsis in mice model, and we have 3 groups, as you can see in the picture in the right corner. And in the middle, we have a septic group, and on the right side is a group which was treated with DNAs. And as you can see, the survival of mice was better after the treatment of DNAs. So this is the way how I would like to break the wall of sepsis.
Marek Schnitzer is the second place, won the second place for 3D printing in medicine. Marek.
So good afternoon, ladies and gentlemen. I'm coming from Technical University of Košice, and I am very glad that organizers make some kind of attempt like this, and I can present some innovations and new ideas of our research and development. We at Technical University, we are involved in medical 3D printing, and nowadays we are working with one project. It is a palate cleft deformation therapeutic process, where we are trying to make preclinical palate cleft plate, and we are trying to dismiss the conventional method because there are 3 problems with the conventional method. It's late, so mother cannot breastfeed the child. It's uncomfortable, so as you see at the picture, when the child needs to have an impression method, it's awful and very uncomfortable for him. And also the precision of the plate which is made by conventional method is very imprecise. So we've developed in many cases in our 3D implants a chain where we could, from obtain data, produce an implant. But my work was this very specific palate cleft device, which is made from a reference model. I produce a reference model in a CAD software where you can manage the data and edit them. And then we have development stadiums, and I put the bioorganic design for silicon coating, because the silicon coating is some kind of something like soft tissue in an oral cavity. You have to look that the child has a very big hole inside the nasal cavity, so we need to be breastfeeded to fill it immediately after delivery. So this is immediately after delivery, 2 hours. Now Simon has the medical device at home. He's drinking tea or milk from an original bottle. He's not using the Haberman one with the big nozzle. And I need to say, because our lawyer said that I need to say it, that it is under a patent appliance, and to be here at it. So that's it. Thank you.
Everybody's the winner, but there are 3 really the final winners. And the 3rd place in this Falling Walls Lab Slovakia competition, the jury decided goes to Katarina Janšáková, using saliva.
Dear colleagues, this high-tech instrument is a device of our daily need. But unfortunately, for some of us, using it 10 or 20 times a day is a routine. I'm talking about lifelong autoimmune inflammatory bowel diseases, and Crohn's disease is one of them. Despite that this disease is accompanied a lot of signs and symptoms, Crohn's disease is very difficult to diagnose, and whole diagnostic process is a long-lasting, painful, and scary torture, which fails very often. Early diagnostics is almost impossible, and some patients are diagnosed too late when the resection of any part of the intestine or even a stoma is needed. And now it's quite clear what's wrong with this patient. As you can imagine, fighting Crohn's disease is a real battle. But the question is, how can we treat these patients properly? How can we put the resection off, at least for a while? First, we need to diagnose them. For this purpose, saliva, which is not routinely used in the clinical practice, can be a very useful tool. Its collection is non-invasive, and you are able to spit out a whole cocktail of markers which can be detected quite easily. And that's exactly what we did. Based on our research, we have found some screening markers based on which we have been able to distinguish healthy individuals and patients with Crohn's disease. So, finding more markers for screening and monitoring of Crohn's disease could save the time between the onset of this disease and its first treatment. So, just spitting in the tube like this means breaking the wall represented by the pain and fear from invasive diagnostic approaches. Thank you.
I would like to welcome here our special guest, Professor Max Gassmann from the University of Zurich, the director of the Institute of Veterinary Physiology.
Today I was said to say something about science, why it's fascinating, why do we do that, and why, and this is especially focusing to the younger people here inside. I mean, and I'm going to talk a little bit, and I have to include a little bit of my life, but I want to basically give some ideas to the younger people here, because we really had very fantastic presentations really today. So thank you very much for your attention. That's what I wanted to say today. Thank you.
I had the chance to see all the 16 presentations, and I was really impressed. And it is for me a big honor to give these 3, vouchers to the winners. And I would like to wish our winner, because she destroyed so nicely the DNA here on the stage, that in the same way you will destroy all the competition in Berlin. Thank you very much.
You will also travel to Berlin, and you will observe how to present and you will have a chance to represent Slovakia.